Hello to everyone. I'm Jakub Knurek and I represent Mabion, a European CDMO specializing in end-to-end biologics development. I feel very sad that I couldn't fly to Hyderabad and meet you personally, but it's a pleasure to be able to present my topic to you remotely during the second Global Biologics India conference. Today I will be sharing insights on one of the most critical components of biosimilar development, the Comparability and Similarity testing panel, and specifically how we define and evaluate critical quality attributes. Let's go through the agenda. We will begin by exploring how CQAs are defined and why they matter so much in biosimilar programs. Then we will walk through regulatory expectations that guide our comparability strategy. And finally, I'll share how our ICH-aligned analytical panel ties into the global biosimilar development plan that meets both scientific and operational needs. Okay, so let's start with the role of the CDMO in biosimilar development. On that slide, you can see the broad picture of drug development requirements for biologics and biosimilars as well. I have added information about service providers who at various stages of drug development can support it with their expertise. As you can see, every stage can be outsourced. How often do pharma companies use that solution? Well, very often. According to industry reports, at some stages more than half of biosimilar drugs pass through the hands of contractors. As you move through the development life cycle from drug discovery to regulatory approval, you see increasing level of outsourcing, with CDMOs becoming essential partners, especially in clinical and commercial manufacturing, but also in analytical drug characterization and regulatory dossier preparation and application. This integration reduces risk and time to market, while leveraging deep technical capabilities and regulatory experience. Now, let's talk a bit about where CDMOs like Mabion fit in this pipeline. As the industry continues to evolve, more and more companies, especially biosimilar ones, are turning to specialized partners and for biomanufacturing support. What you are seeing here is a representation of how responsibility shifts from in-house to outsourced as a project progresses toward commercialization. CDMOs can often accelerate timelines by offering plug-and-play capabilities. For example, proven analytical methods, ready-to-run GMP facilities, or experienced regulatory team. That means instead of building infrastructure or developing methods from scratch, the partners can focus on strategy while we handle the heavy technical work. And for biosimilars, where time to market is everything, this model becomes a huge competitive advantage. You know what also matters in the globalized environment? Positioning. Mabion operates from Central Europe, an emerging hub for biopharma manufacturing and logistics. Our location enables rapid access to EU market and streamlines global supply chain operations, especially important when scaling biosimilars for regional launches. Here's where our capabilities really come into focus. We operate 2 core facilities in Poland, a GMP certified manufacturing site in Konstantynów Łódzki, our headquarters, and the GLP certified analytical laboratory in Łódź for the clinical purposes. Together, these facilities provide end-to-end solution for biologics. And crucially, these services are all aligned with regulatory standards. Our quality systems have withstood global regulatory scrutiny. We've been regulatory audited for both ISO, GLP, and GMP compliance, with no critical findings across inspections. This high level of trust in our systems gives our customers, and of course regulators too, the confidence that biosimilars developed with Mabion meet the highest quality standards. So I mentioned regulators. Yeah, let's zoom in for a second into this matter. So, in reality, development often begins 2 or maybe even 3 years ahead of expiry of the originator patent by early analytical characterization, reference product sourcing, and reverse engineering. You know how it is. Each phase builds upon the previous one, with a strong emphasis on analytical evidence early on. So, regulators, oh, sorry for that, regulators from ICH, FDA, and EMA all agree that analytical testing is the foundation of any biosimilar development program. That's how regulatory guidelines said. That's because these analyses provide the first and often the strongest evidence that a biosimilar is highly similar to its reference product, long before any patient is dosed. Perhaps what I'm about to say will be politically incorrect, but I believe that precisely because of the bureaucracy, typical review schedules are currently more than a year, and only if there are no serious objections to the results presented. This brings me to the thoughts one of my mentors, Ivo Abraham. The guidelines did not loosen the reins. They changed the track. So, yeah, further regulatory changes, including things currently happening at the FDA, may introduce new paths for pharma. They shift the focus. Instead of requiring large, costly phase 3 clinical trials by default, the emphasis is now on demonstrating biosimilarity through deep analytical and functional comparability. So, it's a change in philosophy from repetition to justification, where smarter, earlier evidence can lead to faster approval, provided it's built on a robust foundation of critical quality attributes. Critical quality attributes? Yeah, that's our king and that's our main topic of today's speech. I will stop here and want to present you with a complementary analytical panel, the implementation of which will ensure the approval of biosimilar drug even in an uncertain regulatory landscape. First, however, let's ask ourselves, why are CQAs needed? When developing a biosimilar, we don't have access to the originator's cell line or manufacturing process, so what we do have in the final product? And that's where critical quality attributes come in. These are the measurable properties of a biologic, like structure, function, purity, and stability, that directly affect clinical performance. That's crucial, both for the companies, for the industry, and for the patients. And also that's crucial for regulators. They expect us to identify which of these attributes are most critical, and then use sensitive analytical methods to compare them head-to-head with the reference drug. The closer the match, the stronger the argument for biosimilarity, and the more confidence we give regulators that the product will behave the same in patients. CQAs help us shift from the clinical data, that's heavy approach, to more efficient, data-driven analytical strategy. When we build the comparability panel, it's based entirely on these critical quality attributes. Each one is mapped to a specific category defined by ICH Q6b, like primary structure, glycosylation, change variants, or biological activity. And for each category, we apply orthogonal analytical methods to ensure we are seeing the full picture. In my opinion, the best proposals are already waiting on the following slides. Let's start with the primary structure. Here, we are confirming that the amino acid sequence of the biosimilar is identical to that of the reference one. Even a single substitution can alter structure, function, or immunogenicity. I think that peptide mapping using LCMS is a gold standard here. It allows us to verify sequence and detect any post-translational modifications, like oxidation or deamination. It's also cheaper and easier to use than an NGS. Next one is the higher order structure analysis, which refers to how the protein folds and maintains its three-dimensional conformation. This is critical because folding affects stability, binding, and biological activity. To assess this, we use circular dichroism to looking at overall folding architecture. That's measured absorption, but that's the tech stuff. I would like to go further to the next point, and I see I'm almost out of time, so I'm skipping to the glycosylation profile. And glycosylation can affect everything from half-life and solubility to immunogenicity and Fc-mediated functions like ADCC. That's why we use multiple analytical approaches here. Any differences must be justified. So glycosylation analysis is both highly technical and strategically important. Okay, next point, the purity, or rather impurities determination testing. It's about detecting and quantifying unwanted variants, whether they are related to the product itself, like aggregates or fragments of, for example, antibodies, or to the process, like host cell proteins or DNA. The presence of even small amounts of certain impurities, and I mean here, for example, endotoxins or residual protein A, can trigger regulatory concerns, so this assay must be validated and really sensitive. Measuring the amount of protein may sound simple, but in biosimilar development, it needs to be highly precise and reproducible. We apply several techniques for this depending on context. For example, ELISA assays when we need antigen-specific detection, but rapid UV analysis for general total protein concentration. Then, this is where we prove that the biosimilar doesn't just look the same, it acts the same. So, biological activity testing confirms that the mechanism of action is preserved, especially for monoclonal antibodies with immune effector functions, but as well for the biosimilars, as well for the other biologics. These results are often placed in the Tier 1 CQA category, meaning they must meet strict equivalence standard because they directly influence therapeutic function. This is also my favorite research, to be honest, since it's where I started working in Mabion. A few years ago, while starting as a biotechnologist, I was involved in analyzing the biosimilarity and comparability of monoclonal antibodies in the clinical trials, and I doing apoptosis and ADCC PBMC assays. So yeah, that's biological activity analysis is still in my heart. Last but not least, at the end of this panel, stability and release testing. So yeah, which that analysis ensures that the biosimilar maintains its quality over time and under real-world condition. And I see this data fit directly into shelf life claims, release specification, and overall product robustness. We could also add here several stories about force degradation, about the freeze-thaw studies, but go to the final thoughts. And to sum things up, critical quality attributes are more than just the technical checkpoints. They are the scientific foundation for demonstrating biosimilarity, So yeah, indeed, if there's one takeaway from today's session, it's this. Regulatory agencies want clarity, consistency, and comparability. And that's exactly what this CQAs panel delivers. Thank you very much for your attention. If you have any questions, please mail me at j.knurek@mabion.eu. Have a great rest of day. See you soon at the next conference.